All right, so welcome everyone to today's webinar. Good morning or good afternoon, depending on from where you're joining our session. So during today's webinar, my colleague Tessa will be talking about 3D cardiac microtissues and their applications in drug discovery. But before she does, I wanted to take a couple minutes to talk to you about Encardia, introduce you to our company. My name is Leonie. I work as head of marketing at Encardia, and I'll be your host for today. Yeah, so Encardia has a great expertise in iPSC technology. iPSC stands for Induced Pluripotent Stem Cells, for those of you who might not be aware of the technology. And we use iPSC technology in drug discovery to help therapeutic developers make their decisions faster, with more confidence, and really help them accelerate their drug discovery and development process. And we do that in a number of steps. So our work starts with sourcing and reprogramming of the cells, which is of course always based on these induced pluripotent stem cells. And after the sourcing and reprogramming, we use the cells in our manufacturing process development and our scale up. Our team has more than 10 years of experience in developing cell types at scale. We have certain well-established manufacturing processes. We're also not new to creating new processes depending on what our customer needs. So these manufactured cells are then differentiated to the disease-relevant cells and then characterized for lineage marker expression by RNA and protein analysis. And then lastly, the cells are used to develop custom assays, depending on what the customer needs, to create disease models and to do specific screenings. So to give you a bit of a better idea of Encardia's capabilities, we have, as I just mentioned, different processes that are being developed and that are well established. Our focus is mostly on cardiac and neural cell models. And yeah, as you probably know by now, the focus of today's presentation will be on this upper part on the cardiac cell models. So these established cell models are used to create disease models based on whatever is needed for a project. We have different ways of working to get to the exact disease model that a customer might need. They can be personalized by CRISPR or by overexpression. They can be chemically induced, or they can be created using physiologically relevant co-cultures. And then after, either the healthy or the diseased cell models are used to perform a wide range of in-house assays, of which you can see a small section here in this overview. Yeah, so as Encardia, indeed, the work that we do is based on iPSC technology. We see that has a number of benefits, and that's on the next slide. Let's see if it works now. Oh no, I think it's missing a part. If you can help me go back one slide, Tessa, thank you. So everything we do at Encardia, it all feeds into our one mission. We truly believe that using human stem cell technology helps bring better therapies to patients faster through iPSCs, and that's something that we'll get into a little bit deeper in today's presentation. So to talk about what my colleague Tessa is actually going to present today, she'll be talking about 3D cardiac microtissues and their applications in drug discovery. So Tessa has been working with us since 2015. In 2019, she decided to pursue a PhD in a collaborative project between the Leiden University Medical Center and Encardia, and that's what we'll be focusing on a bit more today. At the end of the session, you'll have time to have your questions answered. So if you have any questions during the session, please ask them in the chat, and we're happy to address them at the end. So Tessa, over to you.
Thank you, Leonie. Do you hear me well?
Yeah.
Yeah, perfect. Thank you for a very kind introduction, and welcome everyone. I'm really happy to be here and to talk a little bit more about our collaborative project with the Leiden University Medical Center, about the cardiac microtissues, how we develop them and how we use them in different kind of assays for safety assessment, disease modeling, and drug discovery purposes. So during this webinar, I will start first with a short introduction about what the 3D cardiac microtissues are, and then I will present about the drug response predictivity of these tissues, followed by how we use them in disease modeling. The last few slides of this webinar will be about how we automate the process of forming microtissues. So in general, we aim to develop cardiac models that lead to a more mature state. This because we would like to have models that can provide us with physiologically relevant information. And I'm sure many of you know a lot of kind of possibilities how to mature a model more. And one of these possibilities is to develop the cells or culture the cells in 3D. An example of a 3D model are these so-called cardiac microtissues. So these cardiac microtissues consist out of endothelial cells, cardiomyocytes, and cardiac fibroblasts, and they're all derived from human pluripotent stem cells. As recently published by Giacomelli et al., they have shown that these cardiac microtissues really enhance the maturation via the tricellular crosstalk. And very interestingly is that also the importance of the non-cardiomyocytes was shown and seemed to be crucial in revealing certain disease phenotypes. Of course, with great opportunities always comes great challenges. So working in general with 3D models, we can still suffer from the variability of these models, the heterogeneity of these models. How predictive are these models really if you compare them to conventional or 2D models? Can we scale them up? What about the throughput? So a lot of these questions or challenges formed actually the basis of this current collaborative project between Encardia and the LEMC. So first about the formation of these cardiac microtissues. So we have established differentiation protocols that are initially small molecule based for the mesoderm induction of all the 3 different cell types. So they're all derived from human-induced pluripotent stem cells, and we made the cardiomyocytes, Ipscs, and FBs accordingly. We freeze the cells, and when we use them for the formation of the cardiac microtissues, we thaw them, and after a pre-culture of either 7 days for the cardiomyocytes or 4 days for the Ipscs and FBs, we combine them in a very specific ratio, and we culture the cells, we plate the cells in 96 well transparent V plates where the microtissues are formed in due time. So usually we maintain them around 3 weeks before we use them for any downstream assays. In our case, for the current project, we are interested in the scalability of these microtissues and if we can use them for more high throughput assays. So that's why I transfer these microtissues to 384 well black plates and use them for instance, calcium. So here we have an example. Let me show you. So we have been developing optimizing protocols for the assessment of calcium transients as well as voltage transients based on the dye. So we can assess the calcium as well as the voltage of a whole plate at the same time of all the different kind of wells using the FDSS microcell system. So the reason why we have picked calcium initially is that it's because a very relevant surrogate to assess effects both on the contractility of the cardiomyocytes or the cardiac model, as well as the electrophysiology in most cases. So here I just included 2 bar graphs where you can see that the beat rate is very stable between the 20 and 25 beats per minute of these microtissues. And the intraval variability, which is in this case, the intraval variability from peak to peak of the calcium transients within one well, is far below 10%, which is our cutoff criteria. And this data is coming from 298 different kind of microtissues derived from 4 different experiments. So I just would like to show you that these microtissues are very reproducible and stable. So now that we have assessed or developed different kind of readouts, we were interested if these microtissues are good in predicting certain drug responses. So for that, we asked ourselves the question, what is a predictive model? And if we follow literature, it's pretty clear that a predictive model should be a model that replicates both the morphology as well as function of the cells or tissue that you would like to study in order to sufficiently demonstrate the physiology or pathophysiology. But we are all, or most of us here, are working in preclinical research, and we are very much aware that depending on your research question, you need a certain model, or your model might be fit for purpose. it could be that you need a model that needs an increased maturity state in order to pick up certain effects. So we were interested in that. And for the current study, we thought the predictive model that we have now, the model, should be capable of detecting certain cardiotoxicities or revealing certain disease phenotypes that we would have otherwise missed in our conventional 2D models. Since we were looking at calcium and we are interested in contractility and calcium handling, we looked at a subset of compounds, positive and negative anatropes, that have a different kind of mode of action on the contractility of cardiomyocytes. So we have here a schematic form where I just would like to notify you that, of course, these kind of compounds, they act differently. They can have an effect on the contractile behavior of the cells, but via different mechanisms. And many of you working with cardiac models know that certain compounds are very easy to pick up. For instance, nifedipine. It's an L-type calcium channel blocker, which is very well developed in any IPS model, and we can measure the effect easily. There are, however, certain mechanisms that are more tricky, so to say. For instance, compounds that are affecting the inner calcium storage or the calcium-induced calcium release. Those compounds can be missed in less mature models. So we were interested in all of these compounds, and we did therefore a blind screen of these 12 reference compounds. And we based our classification of being a positive inotrope, negative or non-effect compound on 7 different calcium-related parameters. And this has been conducted by 2 independent operators. So I jump immediately to the conclusion of this blind screen. So as you can see here, this is the column where we filled in our predictions, so either positive, negative, or non-effect, and after unblinding what the actual classifications was. So we see here a result of a 92% success rate. So we were really happy with this result. But we were also interested in what is the differences. So how the differences between, for instance, the 2D model and a 3D model. So if we pick out one compound that is known to be a bit of a tricky compound, namely levosimendan, it is a calcium sensitizer and also PDE3 inhibitor. And if you look closely at how the 3D model and how the 2D model responded to this compound, we see that both the models are perfectly capable of measuring a positive chronotropic effect. So that means an increase in beat rate. But if we then look at the amplitudes, we see that only the 3D model is capable of showing a positive anotropic effect, which is really what we want to see with this kind of compound. So, excuse me, for these kind of compounds, for this setting or this research question, people might want to go for a 3D model rather than going for a 2D model. From safety assessment, I would like to move forward to disease modeling, where we also use these microtissues for. So one of the diseases that we are interested in here at Encardia and at the LEMC is CBVT1. And I would like to make clear that this is just one example. At Encardia, we have access to different kind of patient lines, or we can modify genetically modified lines if we have you have a certain mutation of interest to look at. CBVT is an inherited and highly malignant channelopathy, which may lead to sudden cardiac death. It is related to the mutation in a ryanodin receptor, which lead to leaky receptors. So what can happen, for instance, with a patient suffering from CBVT is that upon certain stress, you get elevated pKa levels that leads to the phosphorylation and subsequent activation of certain channels like the L-type calcium channel, phospholambin, and that leads to an increase in calcium both in a cytosol as well as in a sarcoplasmic reticulum. This kind of calcium overload is then leading to calcium spillover in a cytosol because the ryanodin receptors are not working properly. And this kind of calcium spillover leads to arrhythmia, as we can see here. For us, this is a perfect phenotype, so to say, because we are working towards using microtissues in high throughputs. We are screening multiple plates at the same time, so we would like to have a very clear black and white phenotype. So seeing arrhythmia would be bad, and seeing no arrhythmia would be good in this case. So we were wondering if this is possible to model in our microtissues. So here we see calcium transients of microtissues that are made with a CBVT cardiomyocyte line. And upon triggering, we indeed see this very heavy arrhythmic behavior of these microtissues that we didn't see in any control of wild type line. And of course, we would like to be sure that this kind of arrhythmic behavior is related to the ryanodine mutation and not a potentially secondary effect or something else, an effect on the genetic background, for instance. So that's why we also treated the same microtissues that showed this kind of arrhythmic behavior. We treated those with flecainide. And flecainide is a well-known drug that is given to CPVT patients for rescuing the arrhythmic phenotype. It's blocking, besides the sodium channel, also the ryanodine receptor. However, flecainide is not always working in the CPVT patients. Certain patients don't respond to flecainide, so this is important to keep in mind. First, I would like to show you the quantification of this study, where we really see that if we trigger these cardiac microtissues, CPVT cardiac microtissues, we do see around 80% of the microtissues showing arrhythmia. And if we treat this with flecainide, we see that all the arrhythmic events disappeared. And I just briefly mentioned that we don't see this with every patient. So that's why we think that this kind of study really paves the way for personalized medicine and finally patient stratification strategies. Imagine to have different kind of patient lines showing severe or mild phenotypes in the clinic. How do they respond? How can we model the microtissues? How do they respond to flecainide or many other kind of compounds? So for us, it will be really interesting to use multiple lines from different patients or multiple corrected lines and different kind of mutations. But of course, for these kind of studies, we have large screens, we use many plates, we need many compound libraries. So at some point, it's not doable to do this manually, like the way I showed you in the first slides. So that brings me to some of the final slides of today, which is moving towards automation. So at Encardia and at LMC, we have developed and optimized different kind of scripts to form, maintain, label, and also transfer the 3D cardiac microtissues for high throughput screening purposes. And we use therefore the TICA and Fluent robots, as you can see here in the video and here in the schematic overview. So what we do is we still pre-culture the cells, as I mentioned before, but then we give the cell suspension, so to say, to the robot. The robot then forms the microtissues directly in the 384 well plate or another plate of interest. So no need to transfer the microtissues, no need to manually seed the cells or to maintain the cells manually. And even if you would like to use another plate at the end, you can always transfer the microtissues also using the robot. So far, I've showed you some data about calcium, some examples with measuring voltage. We also looked at biomarkers. We are currently looking into high content imaging of these microtissues, but you can imagine, of course, that there are many possibilities to do functional analysis or structural analysis, gene expression. And then the final slide, I would like to show you just some results where we show the similarities between the manually seeded microtissues compared to the automated microtissues, because of course we would like to be sure that the robot is making exactly the same kind of microtissues of same quality. So we did a volumetric assay where we show an inter and intra variability of volume that is below 5%. And if we use the microtissues in a calcium assay, we see that the intraval variability of both is below 10%, and the automatic microtissues showed an even lower variability compared to the manually seeded ones. We also looked at the viability and also performed some compound responses. And as you can see, the results are very similar between the automatically formed microtissues compared to the manually seeded microtissues. So the quality is more or less similar, but the biggest advantage is, of course, that we just spend minutes instead of hours and hours in the lab, and not even talking about a potential human error that can happen always, of course, when working with these kind of advanced models. So a very brief and quick overview, but I hope you have learned today that these kind of 3D cardiac microtissues that we develop here are scalable and reproducible. They are amenable to automation and show a very high regularity as well as predictivity for cardiac safety assessment. The 3D microtissues can also reveal a relevant and a rescuable disease phenotype. And we believe that these results that are presented today really show great potential for the large-scale application of physiologically relevant 3D cardiac disease models in high-throughput screening during early stages of drug discovery. With that, I would like to thank you all for your attention, and the Leiden University Medical Center, and specifically Christine Mamry's lab, and all the great funding that we got that made this work possible. With that, I think I will give the mic back to you, Leonie.
Yeah, thank you, Tessa, and thank you for your great presentation. It's also always super exciting to hear you talk about your work. I think there's great potential there, so that's really something amazing. So we would like to open the floor also to you guys, to everyone that's on the line. If you have any questions, please add them to either the chat or to the Q&A function. Tessa is happy to answer them. And we received some first questions to get us started. So one of the questions is about, so what about the cost and complexity if you compare your 3D models to 2D models?
That's a good question. I think in general, the field thinks that, thinking about 3D means complex and a lot of material, a lot of expertise and cost and time. That's why we focused here on a relatively simple 3D model that requires just a very few cells per well. So regarding cell material, it's only 5,000 cells per well. So that's a very cost efficient, I would say, and not complex and easy to transfer. They are not vulnerable. If you have monolayers in a 384 well format, they can be quite vulnerable. So in that sense, I would say it's easy and not as expensive as people might think.
That sounds really good. So over to the next question. Did you use the Giacomelli protocol to differentiate iPSCs into the 3 different cell types?
That's a good question. So we actually use that protocol as the basis protocol, but we have optimized it, especially the initial mesoderm induction step, so that we make use of small molecules. And that reduces the variability you have if you are only dependent on growth factors, for instance. And for the cardiomyocytes, that has been published as well. For the other 2 cell types, that's yet to come.
All right, thank you. So for the next question, is there a key biomarker for each cell type that you always measure for quality control?
Also a good question. I think for we do perform FACS or immuno for the different cell types that we differentiate. For cardiomyocytes, that can be cardiac troponin T or alpha-actinin, together with MC2V, for instance, if you are interested in the ventricular phenotype of the cardiomyocytes. For the endothelial cells, it can be CD31 or 34. So yes, we do use different kind of markers for FACS and or immuno assays.
That's great. Thank you. The next question, do you observe necrotic points in your microtissues?
Necrotic what?
Necrotic points.
Points. I do think it's a relevant question. I did not observe it. I'm also paying a lot of attention if I'm using, for instance, certain dyes or markers that they go through the whole tissue and you don't see any signs of non-beating areas in the middle or necrotic sites in the middle. I think if you go too large in your cell size, so these microtissues consist out of 5,000. I think if you go over 10,000, although I've never confirmed it, I do believe that you need to be careful that the inner part of the microtissue is still viable and not necrotic. So I think it's a relevant point because I think it could happen. But so far, luckily, never observed in the microtissues from this scale.
That's great. Yeah, so far so good then. It's a really good sign, I'd say.
Definitely.
So maybe you already mentioned it during the presentation, but one of our attendees is asking, what is the efficiency or the success rate of producing the microtissues with the TCAN robots, or using the microtissues rather?
The success rate in one go or over different, in general, it's very, I mean, if the difficulty lays in the optimization. So we do the programming, the dry test, the liquid test. So now we have the scripts fully optimized for this kind of procedure, and then the success rate is 100%. If you look at performing different assays downstream, then the success rate can become lower. For instance, if you are working with a voltage dye and you require different kind of washing steps, you of course want to remove as much as media as possible, but you don't want to lose the microtissue. So you need to find a balance, which can lead to not a success rate of 100%, but maybe 95%. I hope I answered the question, but regarding formation of the microtissues, it's 100%.
Okay, that's great to hear. I hope the question was answered to your liking. If you have any additional questions, please type them in. To go to the next question, there's a lot of them in here. Thank you so much for all of your interest. I think we have time to answer 2 more questions. So one of them is there any insight on incorporating the cardiac microtissue on immune-based cardiotoxicity?
Also a nice question. So for the current project that we have here, not so much, but there are already people working on indeed co-culturing cardiac microtissues with different kind of immune cells, such as macrophages. It is also really interesting to see if the microtissues itself are already immunopotent. So quite some preliminary work is being done at the moment, also at the department at the LAMC. And so I think there are a lot of possibilities, and I would say, and Leonie, fill me in, if there are specific interests for Ncardia doing this kind of assays, then we can definitely talk about it.
Definitely. Yeah, I fully agree. There's, I think, as mentioned, there's great potential in using these cardiac microtissues, and we can use them for a broad range of applications, I'd say. So this is perfectly in line with that. And then to go to the final question, again, thank you everyone for your attention, for asking these questions. Unfortunately, we're running out of time, so just to do one more. So how long is the differentiation protocol for your cardiomyocytes, and when do you thaw them? How long does it then take for them to start beating again? So that's 3 questions into one.
The cardiomyocytes, from stem cell to cardiomyocytes, it can be between 2 and 3 weeks, and after day 9, 10, you would definitely see the cardiomyocytes popping up and beating. After thawing, it is the first contractions usually appear within 24 hours. Then they start to formulate this network, so you see like a little bit of slower or no beating, and then in 2-3 days they fully contract. If you then dissociate them and use them for microtissues, then they will beat the next day usually.
Okay, thank you so much, Tessa. And thank you everyone for your attention, for joining today's webinar. Thank you for all your interesting questions. I hope you enjoyed the session. So after this presentation, we'll send you the on-demand version. So if you want to watch again, you definitely can. If you do have other additional burning questions, please feel free to contact us. You can do that via support@ncardia.com. And Tessa will definitely, or someone from her team, get back to you to give you all the information that you need. So thank you so much, and have a nice day.
Thank you all.